In previous videos, you've explored the processes of transcription, translation, gene regulation, and how this knowledge shapes modern science. Now let's put your understanding to the test with a few questions. Question 1. The diagram shows the alignment between the complete genomic DNA sequence of a gene and the corresponding processed mRNA sequence. The upper strand represents genomic DNA and the lower strand represents processed mRNA. Vertical lines indicate identical nucleotides. Gaps represent sequences present in DNA but absent from the processed mRNA. The circled lowercase a, or alias sign, indicates the 5' guanosine cap. The numbers at the end of each line of the nucleotide sequence represents the total length of the sequence to the end of that line in nucleotides or bases. A. At which nucleotide position does transcription begin? Answer: position 206. This corresponds to the first nucleotide present in the mRNA. B. At which nucleotide position does transcription end? Answer: position 675. This position corresponds to the final nucleotide that is complementary to the processed mRNA sequence. Up to this point, the mRNA is directly transcribed from the DNA template strand. Beyond position 675, the stretch of adenines present in the mature mRNA does not correspond to bases in the corresponding position on the DNA template. Instead, this region represents the poly-A tail, which is added post-transcriptionally during mRNA processing. The poly-A tail is therefore not encoded directly in the DNA sequence, but is enzymatically added after cleavage of the pre-mRNA. C. At which nucleotide position does translation begin? Answer: position 284. This corresponds to the A of the first AUG, which is the start codon in the mRNA. There can be other AUG codons present in the sequence. D. At which nucleotide position does translation end? Answer: position 596 till 598. This corresponds to the first in-frame stop codon after the start codon. A stop codon, UAA, UAG, or UGA, terminates translation only if it is in the correct reading frame established by the first AUG. Stop codons do not encode an amino acid. E. How many introns are present in this gene? Answer: 1 intron, identified as the internal DNA sequence absent from the processed mRNA. F. What are the nucleotide positions of the intron? Answer: nucleotides 354 till 464 in the genomic DNA. G. How many exons are present in this gene? Answer: 2 exons. 1 intron separates 2 coding regions. H. How many amino acids are encoded by this gene? Answer: 67 amino acids. There are 67 codons between the start codon and the stop codon. The stop codon does not encode an amino acid. Question 2. When protein X levels are high, X binds to activator site A and induces transcription of protein R. Protein R acts as a repressor by binding to operator region O, preventing RNA polymerase from binding to promoter P1 and inhibiting transcription of genes Y and Z. When X levels are low, X does not bind to A, so R is not produced. Without the repressor, RNA polymerase binds to P1 and transcription of Y and Z occurs. A. Does this schematic describe gene regulation in prokaryotes or eukaryotes? Why? Answer: prokaryotes. This describes an operon structure in which multiple genes are controlled by a single promoter and operator. Eukaryotic genes are not typically organized into operons. B. At different levels of X and R, predict whether you would have transcription of Y and Z in each haploid cell shown. Note that each mutation indicated is assumed to abolish the function of that region completely, that is, complete loss of function. For each mutation, explain your answer. Answer: in mutant 1, the activator is non-functional, so R cannot be induced even when X is high. Therefore, Y and Z are transcribed unless repressor protein is artificially added. In mutant 2, the operator is non-functional, so the repressor cannot bind. Y and Z are transcribed in all conditions. In mutant 3, the promoter is defective. RNA polymerase cannot bind, so Y and Z are never transcribed. Question 3. Which statement correctly describes transcription termination in prokaryotes and eukaryotes? A. Termination occurs when RNA polymerase reaches the stop codon in both prokaryotes and eukaryotes. B. Prokaryotic transcription termination may involve formation of a GC-rich hairpin followed by a run of uracils, whereas eukaryotic transcription termination is linked to RNA processing signals. C. Eukaryotic transcription termination requires the sigma factor. D. Prokaryotic transcription termination requires cleavage of the RNA downstream of a poly A signal. Answer: B. Prokaryotes can terminate transcription via intrinsic hairpin-dependent mechanisms, while eukaryotic RNA polymerase II termination is associated with polyadenylation signals and RNA cleavage rather than a simple hairpin structure. Question 4. What is the relationship between DNA methylation and transcription in eukaryotes? Answer: DNA methylation, particularly at promoter CpG islands, is associated with reduced transcription. Non-methylated DNA may be transcribed, but additional regulatory mechanisms are required for gene activation. Question 5. The following DNA fragment contains a gene's translation initiation codon. The orientation of the template strand for mRNA synthesis must be A. Upper strand 5' to 3' orientation, B. Lower strand 3' to 5' orientation, C. Lower strand 5' to 3' orientation, D. Upper strand 3' to 5' orientation. The answer is B. Lower strand 3' to 5' orientation, because RNA polymerase synthesizes RNA in the 5' to 3' direction. Therefore, it reads the template strand in the 3' to 5' direction. Question 6. What sequence on the template strand of DNA corresponds to the first amino acid inserted into a protein? A. TAC, B. UAA, C. UAG, D. AUG. The answer is A. TAC. The start codon on mRNA is AUG. The template DNA strand is complementary to mRNA. Therefore, the corresponding template sequence is TAC. Question 7. A new antibiotic is designed to bind specifically to the 70S ribosome and prevent peptide bond formation. Which of the following best explains why this drug would selectively affect bacteria but not human cells? A. Human ribosomes are 80S and differ structurally from bacterial 70S ribosomes. B. Human cells lack ribosomes in the cytoplasm. C. Bacterial ribosomes translate proteins at a faster rate than human ribosomes. D. Human cells use a different genetic code for protein synthesis. Answer A. Bacterial ribosomes, 70S, differ structurally from eukaryotic ribosomes, 80S. These structural differences allow selective targeting of bacteria without significantly affecting human cells. Question 8. A mutation prevents the binding of transcription factors to the Tata box. Which process would be most directly affected? A. Transcription initiation in prokaryotes. B. Transcription elongation in prokaryotes. C. Transcription initiation in eukaryotes. D. Translation initiation in eukaryotes. Answer C. The Tata box is a eukaryotic promoter element recognized by transcription factors such as TF2D containing transcription binding protein, which recruits RNA polymerase II. This affects transcription initiation in eukaryotes. Question 9. You are interested in the pattern of expression of a specific gene X in different tissues from the same organism. You isolate RNA from the tissues, run the samples of the RNA in different lanes on an agarose gel blot, and probe with a radioactive probe complementary to exon 2 of gene X. When you perform autoradiography on the blot, you find that the lanes each have a band of a different size and some lanes have no bands. What can you conclude with certainty from the results? A. Gene X is not present in some tissues. B. There is alternative splicing of the primary transcript of gene X in different tissues. C. Within the same tissue, there is alternative splicing of the primary transcript of gene X. D. Gene X does not code for a protein. Answer B. There is alternative splicing of the primary transcript of gene X in different tissues. The radioactive probe is complementary to exon 2, so any detected band represents an mRNA containing exon 2. The presence of bands of different sizes in different tissue lanes indicates that transcripts containing exon 2 vary in length between different tissues. This difference in transcript size is most consistent with tissue-specific alternative splicing. Option A is incorrect because all somatic tissues from the same organism contain the same genomic DNA. Absence of a band indicates lack of expression, or in this experiment, lack of expression of a transcript with exon 2, not absence of the gene. Option C is incorrect because the differing band sizes occur in different lanes, each representing a different tissue. There is no evidence here of multiple transcript sizes within the same tissue. Option D is incorrect because this experiment examines RNA, not protein, and therefore provides no information about whether gene X encodes a protein. Question 10. A researcher identifies a conserved 8 base pair DNA motif located 200 base pairs upstream of gene X. Mutation of this motif abolishes transcription of gene X in liver cells but has no effect in muscle cells. Chromatin accessibility assays show that the region is open in both cell types. Further experiments reveal that a liver-specific transcription factor binds directly to this motif. Which of the following best explains these findings? A. The motif functions as a core promoter element required for RNA polymerase binding in all cell types. B. The motif is an enhancer element that recruits a tissue-specific activator required for transcription in liver cells. C. The motif encodes a short regulatory RNA necessary for transcriptional activation. D. The motif acts as a silencer that represses transcription in muscle cells. Answer B. The motif is located upstream, -200 base pairs, consistent with a cis regulatory element, such as an enhancer or proximal regulatory element. Since mutation abolishes transcription only in liver cells, the motif is not necessary for transcription in all cells and is not part of the core promoter. Also, since liver-specific transcription factor binds directly to the motif when the motif is not mutated, enabling transcription to occur in liver cells, it must be an enhancer binding site recruiting transcription factors specific to liver cells. A is incorrect because core promoter elements like Tata box are required in all expressing cell types. C is incorrect because motifs are DNA binding sites, not RNA coding sequences. D is incorrect since mutation abolishes transcription in liver, not muscle. I hope working through these questions has helped reinforce and clarify the concepts from this topic. If so, do press the like button and support the channel by subscribing.